Good afternoon. Namaste. This is Dr. Uma Keruvetal, Faculty of Agricultural and Veterinary Sciences, Jayoti Vidyapeeth Women's University, Jaipur. As you know, I am the instructor for bioenergetics and metabolism course this semester. And in the previous session, we have looked into the transamination reactions. Now, in the amino acid metabolism, the next part will be the deamination reaction. So transamination reaction happens when like one amino acid is not required at that time, but we require some other amino acid at that time. And then the amino group from this amino acid is transferred to another keto acid, and thereby that can aid in the formation of a amino acid. Now, if the carbon backbone of this amino acid can be used for other metabolic purposes such as gluconeogenesis and all. So in this session, we will be looking into the deamination reactions. So when we have a oversupply of nitrogen in the form of amino acids, so then deamination happens. So deamination is the process that carries out the breakdown of amino acids in such a way that this process releases the free ammonia that is actually highly cytotoxic. So it will be metabolized to urea by the urea cycle that we will be discussing in detail later in the later part of this course. So urea synthesis, which requires a lot of energy, takes place in the liver, and all this excess nitrogen needs to be transported to the liver, and the transport system is actually alanine, glutamine, and aspartate. So these three amino acids accept these ammonia and then transports it to liver, where it will be processed to form urea. So if you were to look into a basic deamination reaction, so it is basically an amino acid which has got an NH3 plus molecule attached, NH3 plus ion attached as one of the side chains. So in the presence of oxygen and in the presence of water, it will remove ammonia, NH4 plus ions, and it will remove H2O2 to give the respective keto acid. So this deamination can be divided into 3 or 4 different types of reactions. There is oxidative deamination, hydrolytic deamination, eliminative deamination. There is also something called reductive deamination. So this, if we were to come into the details of the various types of deamination. So the first one is the oxidative deamination. So in oxidative deamination, what happens? Oxidative deamination is something similar to transamination in that it also in transamination Schiff's base is formed with a so here what is happening is a Schiff's base is formed with a dehydrogenase. Like in transamination, what happens is there is a transaminase enzyme that is involved. Over here, it's a dehydrogenase enzyme. Then this specifically turns the amino group into an imino group. So instead of a C single bond NH3 plus, there will be a C double bond NH2. And then what happens? NH2 plus or C double bond NH, if we are not looking in the ionized form, in that normally occurs in hydrated environments. So then these electron acceptors are the coenzymes NAD plus or NADP plus that are reduced in this process to NADH or NADPH. So then by addition of water, this imino group is converted into an alpha keto group, and along with the release of ammonia. So one of the major enzymes that is involved in this is glutamate dehydrogenase. So if we were to look at the mechanism for this, what happens is this is glutamate with the NH3 plus over here. So when we are adding water, so first what happens is this one, the enzyme will dehydrogenate this one. So one H will be removed from here, one double bond will be formed over here. This H and this H will be removed, and that will form the hydrogen peroxide. And then once that happens, water will be added, and water will make this into double bond O and remove the ammonia. So NH4 plus will be removed. So that is what happens for the reaction of glutamate dehydrogenase. So that enzyme is very important. So we will be looking at the mechanism of action of that enzyme glutamate dehydrogenase as well. So this reaction is like glutamate will give 2-oxoglutarate plus ammonia, and this oxoglutarate will be utilized by the TCA cycle for production of energy, and glutamine, which is the major circulating amino acid, effectively serves as the carrier of this carbons of 2-oxoglutarate as well. So that is also another important points to note, that glutamine in this reaction actually effectively carries the ammonia and the carbon. And glutamate dehydrogenase is not specific. It can either use NAD plus, or it can use NADP plus. So in glycolysis, we had seen that the reactions use only NAD plus, and that will be converted to NADH plus H plus. And in the pentose phosphate pathway, we had seen that it only utilizes NADP plus and reduces it to NADPH. And but glutamate dehydrogenase is not specific in that, and it can use either of the nicotinamide nucleotide factors. And this converts free ammonia and to oxoglut, if it's like accepting ammonia, then it converts oxoglutarate to glutamate, and otherwise it will be removing in the deamination reaction. It will be, so it's a reversible enzyme as well. So that is like there is also this major thing that glutamate, as I've told you, that is one of the major circulating amino acids. So it also acts as the principal amino donor to other amino acids in transamination reactions. It acts as the ammonia carrier to liver where urea synthesis occurs, or it acts as the ammonia carrier to kidneys where ammonia production occurs, and that ammonia acts as the pH regulator, and this ammonia is rapidly removed in the form of urine. So then that is about oxidative deamination. This is called as oxidative deamination because there is oxygen involved in here. Like the oxygen is added to the and the this one becomes the alpha keto group. Now there is the simple hydrolytic deamination. So a hydroxyl group is reversibly attached, and the amino acid is eliminated in the form of ammonia. So the enzyme responsible for this, it's glutamate dehydrogenase. In oxidative deamination, it is glutaminase in hydrolytic deamination. So the reaction, the final end products is similar. So if it's asparagine that is involved and aspartate is produced, then asparaginase is the enzyme involved, and asparaginase is also a very clinically relevant enzyme, and it's used in the treatment of multiple metabolic disorders, and even in some cases for the treatment of cancer. So then if we were to come into the third form of deamination, so that is the eliminative deamination. So in eliminative deamination, small amino acids such as serine or cysteine, it can remove its nitrogen in the form of ammonia by the elimination of water, hydrogen sulfide. Like, as you know, cysteine has got sulfide groups and sulfur in it. So it can actually eliminate in the form of water, hydrogen sulfide, or sulfurous amino acids. So the other sulfurous amino acids can also be formed. And once for this reaction, again, pyridoxal phosphate is the coenzyme that is required. So this hydration actually creates a double bond, and following hydrolysis, alpha keto acid is formed. So the mechanism as such for these three forms of deamination is not the mechanism is different, but the end products are not actually very different, except in the case of eliminative deamination, where instead of ammonia alone, ammonia and water, hydrogen sulfide is formed, and sulfurous amino acids are formed. So that is the major difference between the other two forms of deamination and eliminative deamination. So now if we were to compare the transamination and the deamination reactions. So as you know, transamination is the transfer of an amino group from one molecule to another, especially from a amino acid to a keto acid, like from glutamate. If glutamine and pyruvate are involved, then alpha ketoglutarate will be formed, and alanine will be formed. So that is what there will be the transfer of amino group from one amino acid to a keto acid. Thereby, the amino acid becomes its respective keto acid, and the keto acid becomes its respective amino acid. But in deamination, the amino group is removed, and so we get only the keto acid and ammonia. That is the, so here there are two sets of molecules that are involved. It could be either one glutamine and one alpha ketoglutarate. That is very rarely happening. I mean, why would one glutamine be transaminated to form another glutamine? But say if we want alanine, we have excess of glutamine, then transfer the amino from the glutamine to pyruvic acid, then we'll get alpha ketoglutarate plus alanine. But in the deamination reaction, what will happen is, if we have alanine, we will get pyruvate plus ammonia. And that is the major difference between transamination and deamination. Now, transamination is involved in the synthesis of non-essential amino acids, whereas deamination is involved in the breakdown of excess proteins. So transamination occurs in all cells of the body, whereas deamination occurs in the liver, where the ammonia is readily shuttled to urea cycle. Then the enzyme involved is transaminases, also known as aminotransferases. And the enzyme involved over here is deaminases, but then there are the different enzymes that are the glutamate dehydrogenase is also an important enzyme that is involved in the oxidative deamination process, and the glutaminase is the uh, enzyme that is involved in the hydrolytic deamination. So this results in an exchange process, and this results in an elimination process. So that is again another one. But in both cases, glutamic acid is the main amino acid that is involved in this, and this transamination is reversible, whereas the deamination is irreversible, and that is the another major difference between transamination and deamination. So hope you have understood this. In the next session, we will be looking at the third step, the decarboxylation reactions in the metabolism, the catabolism of amino acids. After which we will be looking at oxidative degradation of amino acids and then urea cycle. So that will be the topics that will be covered under the catabolism of amino acids. So that'll be all for this session. Have a nice day. Thank you.